Hello, and welcome to More Intelligent Tomorrow, a wide-ranging exploration of the potential impact of AI on the world around us, as envisioned by the future heroes of the human and machine intelligence revolution. How do human intelligence and artificial intelligence collaborate to unravel the human genome, and impact the future of wellness and disease through the power of AI? We'll discuss this and more with Dr. Lee Hood on today's episode. And now, your host, R.E. Kaplan.
So welcome, Lee.
Pleasure to be here.
We are extremely excited to have you on as our guest. You have an incredible background, having created the automated DNA sequencer, unlocking the foundations of life with the Human Genome Project, and what you're currently doing now with the Institute for Systems Biology. And also we're extremely interested, based on your perspectives, of what is going to happen in the coming 10 or 20 years.
Well, it's a pleasure to be here talking with you about these very exciting times.
Why don't you tell us about yourself a little bit?
I grew up in Montana in a very small town. My high school had 46 members in my class. I had three of the best teachers I ever had there, and they treated me as an equal. It made me realize that there was a big wide world out there that could be explored. And in fact, one of them even argued that I should go to Caltech. I was resistant for a variety of reasons, but in the end, I did decide to go to Caltech, and it was the best choice I could have made. And I really started thinking about wanting to do human biology back when I was 6 years old, and my youngest brother was born with Down syndrome. And I remember asking the physician at that time just what caused it, and they had absolutely no idea whatsoever. And that really intrigued me and stayed with me a lot of the rest of my life. One seminal event that happened while I was still in high school was my chemistry teacher. This was back in 1956. I taught a lesson on the structure of DNA just 3 years after it was discovered. And what I came away with was this elegant molecule, the basis of life, is something that's certainly worth getting into. And that really decided and sent me into biology. And I've pursued DNA in one way or another since that time. But anyway, back to Caltech. I decided from Caltech to go to medical school. And Johns Hopkins turned out to be a terrific medical school. And in the end, I decided to go back to Caltech and get my PhD because Professor Bill Dreyer had just moved there. And he had done some really spectacular work I read when I was in medical school on studying tumors from multiple myeloma to understand the structure of antibodies and hopefully how they functioned and how their diversity was generated. And that sounded to me like just exactly the kind of topic I wanted to do. When I went to Caltech, that was when, in many ways, I felt my career really got started.
I also went to Caltech, and that's actually how you and I met a little over 30 years ago. I don't know if you remember, but there was this conference of like top student researchers from around the country. And, Caltech had the SURF, Summer Undergraduate Reference.
Right.
And you keynoted, and I had a huge honor to introduce you. And I said, let me introduce Dr. Hood. Since at that point, you had 8 doctorates, the audience loved it. And now you have, I think, 17, if you include honorary.
Right.
Yeah, but then after I graduated, you had asked me, I became proficient in databases to work on the Human Genome Project. But one of my, I don't know if it's a regret or not, but paths in life was I ended up working with Major League Baseball instead of working on that project, but super thrilled that it successfully completed the sequencing of the entire human genome in, was it 2003?
It was 2003 when, the first supposedly complete sequence, but it turns out we still had a ways to go before it was entirely finished. But for the purposes of politicians, in 2003, it was finished, and there was a broad announcement, and it was on the cover of Time magazine and others. It seemed to yield so much promise for the future. And then finally, I had a really terrific student, Roger Perlmutter, who was then chair of immunology at the University of Washington. And he said, "Look, Lee, would you consider coming up to Washington?" He said, "Maybe we could get you a meeting with Bill Gates." And he said, "What I'm going to do is invite you to give 3 lectures here," and they were called the dance lectures, "and give us your view of what the future of biology and medicine is." And Bill went to all of those lectures. So in 92, I picked up and brought my group up to the University of Washington and started this new department, recruited absolutely terrific people from all across the country. And that department turned out to be absolutely spectacular. So Phil Green developed the key algorithms for the Human Genome Project, that is, how to assess the quality of DNA sequence that was generated. I developed there then the 5th instrument that I pioneered, and that was using inkjet technology to synthesize very rapidly DNA. So you could make DNA arrays, or you could make DNA fragments, and we ended up commercializing that through Agilent, and that's the technology they use today for very large-scale DNA synthesis and so forth. But, the department was absolutely an enormous success, but I'd found that the bureaucracy of the University of Washington really made it difficult to get new ideas started there. And I can give you an example. I said, look, if to do systems biology, we really need a lot of computing power because we're going to be looking at a lot of data and a lot of complexity and so forth. And the head of computing at the University of Washington came down and showed me this really small janitor's room in this area I was in. And he said, look, we'll fix this up for you. It's no problem. That's all the computing a biologist will ever need. And so I decided, and that was one of 10 things that I could mention that maybe if I was really going to develop an entirely new field, and systems biology was, I'd have to do it in a different way.
Yeah, one of the things, with our listeners is how human intelligence and machine or artificial intelligence collaborate. And I know one of your big innovations was automation. So automation of DNA sequencing. So where do you see artificial intelligence and human intelligence collaborating?
We have a big artificial intelligence problem that we're just getting started on with this 1,000,000-person project. And we should probably go back sometime and review that. But the idea is in 5 years, we will have the complete genome sequences of 1,000,000 people, and we'll have an enormous amount of longitudinal data that'll include blood analytes, that'll include the gut microbiome, that will include digital measurements of quantized self and all of these kinds of things. And what's interesting as we accumulate these data over this 5-year period, we're going to identify more and more actionable possibilities. And that's going to come about through the integration of different data types in particular. And those actionable possibilities are things that if a patient does them can either improve their wellness or will let them avoid disease. And they're usually validated by going to the literature and finding out, yes, there are these different possibilities. But the 1,000,000-person project, we started a company that brought scientific wellness to consumers, and we probably had about 400 actionable possibilities, both wellness and disease related there. But I'm arguing when we do the Million Genome Person Project, and we had 5,000 people in Airvale, and we'll have 1 million here. That's a 200-fold increase. I think we're going to have thousands upon thousands of new actionable possibilities. And there'll be so many that physicians won't stand a chance of really knowing about them or understanding. We're doing this for Alzheimer's. We have to create a knowledge lake, which basically has all of the known data about Alzheimer's, okay? And then the idea is we can use this knowledge lake in the context of AI, describing it and laying it out and so forth. And it will have the capacity then to take the individual data from an Alzheimer's patient and process it through this lake, and it will output a diagnostic series of recommendations and a therapeutic series of recommendations. And it will prioritize those according to the needs of the patient. And it will explain those so that a physician can look at these things and understand what they're about. What this will make possible is we can make a physician in a small town in Montana basically equivalent to an Alzheimer's expert by letting the AI do the analysis and then feeding it back to the physician in a digestible manner so he can respond to the needs of the patient.
The applications are stunning. So for the 1 million human genomes, is that the full sequence, like the 3 billion
It's the full
pairs?
sequence. We're going to get all 3 billion nucleotides. We developed an algorithm which gives you your biological age, that is the age your body says you are, as opposed to the age your birthday says you are. And the obvious idea is the younger you are relative to your chronologic age, the better you're aging. We looked at 40 different diseases, most of them trivial diseases, that the Airvale patients had. In every single case, their average biological age was well above their chronologic age. So when you're sick, your biological age is higher. And in fact, for diabetes, we had 300 people that were diabetics. Their average age was 6 years older than their chronologic age. It's a metric for wellness, and it's a metric for disease. So what we've also been able to show is we can actually determine the biological age of your major organs, your immune system, your liver, your kidney, your heart, et cetera, et cetera. So you can get this global age, but then you can know what each of your organs are, and we can focus therapies toward optimizing your aging. So my vision for the future is when we're doing longitudinal analyses on all individuals, as we will in the future, we'll be able to see your earliest transitions. We'll be able to use biomarkers to identify the networks and so forth that are engaged in those changes and develop therapies that can reverse the disease before it ever manifests itself as a disease phenotype. And this is what I call predict and prevent. What we've beautifully shown that as a disease progresses, the disease-perturbed networks become increasingly complex. And where contemporary medicine treats patients is after diagnosis. And for many chronic diseases, that's way too late to be able to reverse them in a simple manner. You've got to go back to the beginnings. So that's the essence of 21st century medicine. And the way that we're going to realize it is through this 1 million person project that we're doing with Providence, where we'll gather 200 times as much data as we had for Airvale. And there we estimate we'll be able to see 175,000 wellness to disease transitions, and to be able then to get really good data on knowing signal from noise and the biomarkers that mark the earliest transition. So anyway, that's my big vision for 21st century medicine.
Did you have any insights or studies of how COVID, long-term effects, or how that might be changing, all of the lifespans?
Let me just say, at the beginning of 2020, we started 5,000 people on the Million Person Project just to learn how to do it. COVID came to Seattle in February, and that shut it down, and we'll open it up again this year. And what we did initially was to take 300 COVID patients and compare them against 3 controls where we drew bloods at when you came into the hospital 7 to 10 days later. And then we're looking at the convalescent and the long haulers and things like that. We are going to be looking at the convalescent individuals. And of course, the sobering reality is a fair number of them have long-term effects that can be mental, that can be cardiovascular, be GI, they can be hemonc, all of these kinds of things. And I think the long-term sequelae of COVID-19 is going to be a really grim story. And it may, for some people, go on and on. I have a good friend who got it and said he now has had for about 2 months the worst general arthritis. He said, I've never had arthritis before. And he said it isn't going away at all.
Why do you think COVID is so hard?
I think the answer to that, no one knows. Clearly, what is unusual about it is its ability to attack many different kinds of organs, one way or another, either through its ability to react with a receptor that's present on lung cells and many other tissue cells, or its ability to evoke a cytokine storm that can lead to difficulties in the brain and things like that. So I think we don't understand much at all about the pathophysiology of it. And of course, it's like most viruses, as we've learned recently, it has the ability to mutate.
And what about COVID testing challenges?
Technically, there are really straightforward ways of doing it. We're actually working on a microfluidic device that is going to be point of care. It will be at home. We have the Gates Foundation, I think, interested in it now because it can multiplex. So we could look, for example, at COVID plus flu, which could be very useful in the winter and so forth. But wouldn't it be wonderful if you had a device that cost about $2 and you could get a new test for less than $1, a little insert that you'd put in, and that everybody had it at home. So when we had the next wave, you could quickly, and this uses saliva. So it's really easy to check and everything. So anyway, that's one thing that we're pushing right now. And there are technologies for doing rapid testing, and they just have to get out to the community more effectively and so forth.
So our podcast is called More Intelligent Tomorrow, where we talk about what the world will be like 10, 20 years from now. So what are your thoughts on where the world could be, or what are the possibilities?
Well, in 10 to 20 years, I think everybody would be carrying out this longitudinal analysis. I think we'd be optimizing your health trajectories. I think we'd have you being well through scientific wellness. We'd be modifying your age trajectory, if not reversing it. We'll see. And I think most important of all, we would probably be a long ways toward eliminating many chronic diseases by being able to see them at the earliest inception point and reversing them.
Lee, thank you so much for your time coming on the show. This is incredibly fascinating. Congratulations on your success, and thrilled to see that you're going to continue to dedicate all of your energy to improving all of mankind. So thank you.
Well, it's been a real pleasure talking with you, Ari. I think You have a terrific job. It must be wonderful to talk to interesting people and learn so much new all the time. So it's kind of like my job. I'm learning new things all the time. So neither of us ever need to retire. That's obvious.